these, okay, I will start. Yeah. Can you see the PPT?
Yes, ma'am.
Yes? Okay. Yeah, I will just repeat. Please mute your mic. So I will just show you the structure of the mitochondria. So in the mitochondria, you have inner membrane space, intermembrane space, which is between the inner membrane and outer membrane. And you have DNA here, and the like what you call the inner foldings are called cristae. Then we have ribosomes, granules, and matrix. And here we have ATP synthase particles. So ATP synthase is the enzyme which is actually involved in the synthesis of ATP molecules. So therefore, mitochondria is involved in the synthesis of ATP. That's why mitochondria are called as powerhouse of the cell. Okay, then ribosomes. So ribosomes are involved in the protein synthesis. So ribosomes are made up of two subunits. So in the eukaryotes, we have 80S ribosomes. In prokaryotes, we have 70S ribosomes. So 70S ribosomes are made up of two subunits, 30S and 50S. So 30S is the smaller subunit, 50S is the larger subunit. Then 80S is made up of 40S and 60S. 40S is a smaller subunit and 60S is the larger subunit. Okay, so what is this S here? It is the Swedberg unit. So it is nothing but whenever the ribosome subunits were centrifuged, based on their density, they have settled at various positions like 30, 40, 50, 60, like that. So based on that, they have given this number. But don't think that, okay, 70S means it should be addition of these two. No. So 30 plus 50 you'll have in prokaryotes, right? So these ribosomes, they are present as, they are also previously called as pallid particle, and they are naked ribonucleoproteins. They are not enveloped in some membranes. Okay, they are protoplastic plasmic particles, and they function as a site for the protein synthesis, and they are the protein factories. So here actual protein synthesis takes place, and membrane is absent and freely present in the cytosol, or they are attached to the endoplasmic reticulum. Suppose if they are attached to the endoplasmic reticulum, they are called as, the endoplasmic reticulum is called as rough endoplasmic reticulum. The attachment is through a protein which is called as SRP protein. So the main functions of these ribosomes are protein synthesis and free ribosomes synthesized. So here you have two types of ribosomes, right? Free ribosomes which are freely present in the cytosol, or which are attached to the endoplasmic reticulum. So those which are freely floating in the cytoplasm, they synthesize the proteins which will be secreted outside the cell. Whereas the ribosomes which are attached to the endoplasmic reticulum, those ribosomes synthesize proteins which will transport. That means usually like membrane bound proteins will be synthesized on these ribosomes, and they will be transported to the site of function. Okay. Then the next is nucleus. So a specialized double membrane bound protoplasmic body. So here all the genetic information will be stored. So this genetic information, that is DNA, will be controlling the cellular metabolism and transmission to the posterity. So the transmission of the genetic information from parent to the child. So that is transferred through the DNA, and that is stored in this nucleus. So it's the largest cell organelle, and it is found in the region of maximum metabolic activity. Okay, and it is spherical or oval in shape. So it consists of a nuclear envelope. So the nuclear envelope, it separates the nucleus from the cytoplasm. So this nuclear envelope, so this is present only in the eukaryotes but absent in the prokaryotes. What nuclear envelope? So this nuclear envelope, it separates nucleus from cytoplasm, and this nuclear envelope is made up of lipoproteins, that is lipids attached to the proteins, and trilaminar membrane. So there are various pores which are called nucleopores present on this membrane. So this will help the movement of let's suppose mRNA, so which is transcribed from DNA. So that has to go out of the nucleus into the cytoplasm and reach the ribosome where the protein synthesis takes place. So such kind of movement of molecules is allowed through this nuclear pore. Then nucleoplasm. So nucleoplasm is a transparent, so semi-fluid, like cytoplasm. It's a semi-fluid colloidal substance present in the nucleus. It consists of nucleosides and enzymes, which are required for this transcription and replication process. Then nuclear matrix. So here a network of fine fibrils of acid protein, so which will be functioning as a scaffold. That means for the chromatin attachment, so whatever the proteins required, so that network is called as nuclear matrix. Then what is chromatin? So chromatin is the actual hereditary material, that is DNA, which is tightly packed. Okay, which along with the protein, that is what are the proteins? Histones are the proteins. Then nucleolus. So nucleolus is a naked, round or slightly irregular structure attached to the chromatin. So here on the chromatin, we have a site called nuclear organizer region, NOR. So to that region of the chromatin, this nucleolus will be attached. So here there are 4 components. One is amorphous matrix, granular portion, where you have proteins and RNA in 2:1 ratio. That means 2 folds or 2 parts of protein and 1 part of RNA. And fibrillar portion, where it is made up of nucleonema, then chromatin portion. So this is the principal site for the development of rRNA, ribosomal RNA. And it stores nucleoproteins, that is proteins which are involved in the attached to the nucleus. So here we have 3 types of RNAs, right? tRNA, rRNA, and mRNA. So that rRNA, ribosomal RNA, is primarily synthesized in this nucleolus. So this is the structure of the nucleolus, where we have, structure of the nucleus, where we have at the center we have nucleolus, then the space is called nucleoplasm. So here we have chromatin, DNA, so inner membrane, outer membrane, then this outer envelope is called nuclear envelope, and the small holes like structures, they are called nucleopores, right? So what is the function of this nucleus? So the function of chromatin is, it's the hereditary material. So it consists of genes. So it's genetic information. So all our instructions to maintain our body, that is all the metabolism, their physical appearance, or everything is coded by the genes. Okay. So the genes consist of DNA material. So, so all the instructions for the growth and development of an organism is dependent on this chromatin. Then this nucleus is controls the cell metabolism. Okay, because so all the enzymes, proteins, lipids, everything. Okay, like the synthesis, then degradation, everything is controlled by this nucleus means the genetic material. Then formation of ribosomes in nucleolus, and it directs the cell differentiation. So during the developmental process, so all of us, like, we developed from a single cell, that is zygote. But so you might think that, okay, all the cells of our body, they came from the single cell. So all our body cells should have same DNA, same genes, but why they are functioning differently? See, brain cells function differently. Liver cells function differently. Differently means different proteins are synthesized. The functioning is different. Okay, so they're called specialized cells. So how this like regulation is happening, all this is because of the genetic material. So then replication of nucleus essential for cell replication. So first replication of DNA happens, then only the cell will get divided. Then what are the chromosomes? So chromosomes are nothing but the rod shaped or thread like condensed chromatin fibers. Okay. So they are the hereditary vehicle which store the hereditary information. So here the two halves, they are called chromatids, sister chromatids, and they are attached at the center, which is called centromere. So here you can see this image. They are attached at the center. So chromosomes are of four types based on the position of the centromere. They are called telocentric, acrocentric, submetacentric, and metacentric. Telocentric means if the centromere is at the terminal, not at the center, but at one end, then that is called as telomeric. Oh, sorry, telocentric. Then acrocentric means if the centromere inner to the telomere means a little like not completely at the end, but not completely at the center. It is in between these two positions, then it is acrocentric. Then submetacentric means this centromere is in the submedian. Okay, that means again not completely at the center, but towards the is little towards one side. Okay, then metacentric means the centromere is at the median, that is at the center. So the chromosomes contains a coiled filament called chromonema. Okay. So what are the functions of these chromosomes? So chromosomes consist of actual genes are located on the chromosomes. So all hereditary information located in the genes is carried by the chromosomes. Okay. And it controls the synthesis of structural proteins. So these proteins further help in the cell division and cell growth. Cell growth and cell division means indirectly the growth of the organism. Then it controls cellular differentiation, and the chromosomes can replicate themselves and produce their carbon copies by DNA replication. And they also produce nucleoli, which will synthesize the ribosomes. Okay. So here you can see the telomeres are at the ends. Okay. So telomere, the function of the telomere is while replication, some amount of the chromosome will be lost. If such kind of thing happens, so after some generations, complete genetic information will be lost. So in order to prevent that, the edges are protected by the telomeres. So telomeres are now, the research is going on that telomeres are involved in the aging process. Okay, if there is any defect in the telomeres. Okay. So here you can see in the embryonic stem cells, the telomere is long, but in the adult stem cells, the telomere is short. Okay, that means the telomere length also will be degraded. Again, that is a different aspect. Okay, if you want to discuss, but just remember this. So these chromosomes, they form a link between the offspring and the parent. Okay, and they determine the sex of individual. So we have sex chromosomes like X and Y in human beings. So if the baby is getting X chromosome from mother and another X chromosome from father, then the gender of the baby is female. If X is coming from the mother and Y is coming from the father, then the baby is male. Okay, so in this way, the gender of the baby is also determined by the chromosomes. And by the process of crossing over, they produce variation. So these chromosomes, please mute your microphone. So these chromosomes, there is a process called crossing over. So one more thing is, there should be some variation. Yes, so there should be some variation. See, if even the children, they won't be same as it is like their parents, right? There will be some kind of variation, though they have some features of the father or mother, but still the children are completely different. They look different. So that is because of the crossing over or recombination. So that means a segment of one chromosome will be fused with the other chromosome. So in such cases, new varieties or new variations will be introduced. And don't think that all variations are causing some disease or something. So these variations may be normal. It is only a phenotype. Okay. And the mutations, some mutations may cause some diseases and if they get accumulated. Okay. And if there is any change in the gene chemistry, then the mutations may be produced. Then the next is cytoskeleton. So cytoskeleton is it will give the shape to the cell. Cyto means cell. So skeleton. Skeleton is, as we have in our body. So what is the purpose what is the role of the skeleton? It is giving you the shape or a like rigidity. Okay. So in the same way, even cytoskeleton is giving the shape to the cell. So it's extremely minute, very small, fibrous and tubular structure. It forms structural framework inside the cell. So it has three proteins: microfilament, intermediate filament, and microtubules. So the main function of the cytoskeleton is to maintain the integrity of the cell. So though the cells are flexible, they are not rigid, but they should not lose their shape. Each cell will have its own characteristic shape. So that shape should not be lost. So to maintain this integrity, this cytoskeleton plays an important role and it causes cyclosis. And it is responsible for change in the plasma membrane during endocytosis and exocytosis. I told you what is endocytosis and exocytosis yesterday. So here you can see these are the microfilaments, then intermediate filaments and microtubule structures. So this is how the cytoskeleton looks like. Then flagella and cilia. So both are almost like similar, but flagella is quite complicated than the cilia. So they are fine hair-like movable protoplasmic processes of cell, which are capable of producing fluid medium for locomotion or movement and passage of substances. So flagella are longer in length but fewer in number, whereas cilia are smaller in size, whereas they are more in number, numerous in number. So what are the functions? Is they help in locomotion in flagellate and ciliated organisms for the movement. Then they create current for obtaining food from the aquatic medium. So aquatic organisms like microorganisms to get the food out of that. So especially a current will be created. Okay. So in land animals, the cilia of respiratory tract, they eliminate the particles which are coming into the air. So they will purify the air and can also act as sensory organs and also involved in the internal transport, that is the transport of eggs in the oviduct and excretory substances in the kidneys. So here you can see the um, here you have the upper one. You can see there are some proteins called dynein proteins. We have outer microtubule doublet, that is two microtubules. They will form a doublet, and we have radial spokes which will be connecting. At the central, we have one microtubule which is called axoneme, and there are some arms which are made up of proteins called dynein arms. Okay. And this movement of these microtubules will enable the flagella to movement. Okay. And in the basal body, which is also called as kinetosome, is present. So even cilia also has similar structure. So here you can see the flagellum. So these are the real-time images. So here you have flagellum. So we have microtubules. It's a longitudinal view and have plasma membrane. So microtubule doublets are present here. Okay. So I told you like what are the chromosomes. So they are dense area of centrosomes. So in the chromosomes we have the like whenever the cell division happens. Okay. So here how it looks like. So in the chromosomes, we can see the centrosomes and centrioles. So centrosomes are the dense area of cytoplasm with radiating microtubules. Okay. They contribute for the building of mitotic spindles. That means whenever the cell division happens, so the mitotic spindles are formed which will pull the chromosomes towards the poles. So that mitotic spindles are mainly built by the centrosomes. Then centrioles. So centrioles are the submicroscopic microtubular cylinder-like structures with 9 triplet fibrils. Okay. They will also form duplicates without having any DNA or membrane covering. So this is the image of the cells, how they look like. And this is during the movement, that is cell division, how the spindles will pull the chromosomes towards the poles. So the functions of these centrioles are, so they help in cell division by forming microtubules organizing centers, MTOCs. Then centrioles can be transformed into basal bodies, which give rise to cilia and flagella. So again, these microtubules are involved in the movement. So even here, the spindle fibers, when they are formed, they have to pull the chromosomes towards the end. So that movement is also facilitated by these microtubules. Okay. Then other cell inclusions like vesicles, all these. So they are non-living substances present in the cells. They are called ergastic bodies. Okay, they may be soluble or insoluble state and organic or inorganic in nature. And they are mainly, we can divide them into 3 categories: reserve food, excretory or secretory products, and mineral matter. So what are the different exclusion bodies? Inclusion bodies are melanin. So here they will store these pigments. Okay, melanin is a pigment which is present in the skin, hair, eyes, and meninges for protection against the UV rays. So if the melanin production during like in areas where they have to expose more to the sun, so the melanin production will be more. Then glycogen, which is present in the liver and skeletal muscles for quick energy breakdown. Okay, so the glycogen is broken down to give glucose. So they will give quick energy. Then triglycerides, they are present in the fat cells and they're broken down and they can be utilized to the ATP. So all these are present in the form of inclusion bodies. Okay, this till here are like, this is about the cell organization. So what are the different types of cells? So what is a cell? So what are the different what are the differences in prokaryotic and eukaryotic cell? And eukaryotic also we have plant cells and animal cells. And what are the different organelles and components of the cells? So that we have discussed till now. So do you have any doubts till here?
No, ma'am.
Okay. So now we will continue with the next topic, that is cell division. So this cell division has to happen like in eukaryotes, so in prokaryotes, that is in bacteria, like it has to continuously divide. So they will have some generation time. For example, E. coli will have 20 minutes of generation time. That means after every 20 minutes, the cells will divide. Okay. So whereas in eukaryotes, we'll have cell cycle. So cell cycle, it takes 24 hours. So it has various stages. So here the cell division is nothing but the cell multiplication or formation of new cells from the pre-existing cells. So it has three ways. One is mitosis, mitosis, and meiosis. So in prokaryotes, as I told, it's a simple division. So the cell division is called binary fission. So cell division has two phases. One is interphase and mitotic phase. So interphase means, the term inter means between, phases means aspect. That means it's a gap phase simply, not gap, resting phase. Okay, resting between the different phases. So it's a series of changes that takes place in a newly formed cell and its nucleus before it becomes capable of division again. So that is called as intermitosis. Okay. So in the cell cycle, we have G1, S and G2 phases. So this interphase, it is usually the synthesis of various enzymes, various to check the DNA replication and to check whether it has happened correctly or not. So all these processes takes place in the interphase. Like once it is done, once the DNA is ready and it's perfectly correct, now then the cell has to divide. Okay, so that is again mitotic phase. So in the interphase, we have G1, S and G2 phase. G is the gap. Okay, G stands for gap, S for synthesis. So in the G1 phase, it's the longest phase and it's a post-mitotic gap phase. So because the first stage it is, okay. So here the cell is metabolically active and grows continuously, and RNA and proteins are synthesized. So after G1, we'll get S phase. So in the S phase, the replication of DNA takes place. So whatever the components required for the replication of DNA, all those are synthesized in the G1 phase itself. So here the RNA and proteins are synthesized, the nucleotides, amino acids for histone synthesis, and energy-rich compounds, everything is formed, and cell organelles are increased in their number. Then S phase. So S phase is called synthetic phase. Here the chromosomes, they replicate, and the DNA molecule, they function as a template for the synthesis of new DNA strands, and the DNA content doubles and duplicate sets of genes are formed. New chromatin fibers are formed, and they are also known as invisible phase or M phase. Okay, here also the actually replication is taking. Okay, chromosomes are distributed. So that chromosomes which are formed here, they will get distributed in the later M phase. Okay, here the subunits are of kinetochores are synthesized and the centromeres begin to divide. Okay, all this happens in the S phase. Once the S phase is finished, now the next step is G2. That means once the division has, once the replication has happened, now the next stage is the division, my M phase. So before that, again, the cell has to cross check whether the replicated DNA is correct without any mutation or not. Okay, so this G2 phase, it is the pre-mitotic phase where the synthesis of DNA stops, formation of RNA and protein takes place, and the preparation of cells to undergo the cell division. Okay, this is about the cell cycle. Okay, the overall phases. This is the interphase. So first is G1, where the cellular components excluding chromosomes are okay, because chromosomes will get duplicated in the S phase. So all these are synthesized. In the S phase, the chromosomes are duplicated, and then the G2 phase, the cells double check the duplicated chromosomes for errors, and if there are any repairs needed, that will be done. Once the G2 phase is finished, then it enters the mitosis. Some cells after G1 then enter G0, G0 phase. That is called cell cycle arrest. So especially the neurons of the brain, they enter G0 phase. That's why there will be the regeneration of neurons is not possible. Okay. Then comes the mitosis. So I will just give introduction and I will stop today. So from tomorrow we will continue with the cell division. So mitosis means mitose means thread or fibril. So it's a type of division in which the chromosome will do replicate and they will be equally distributed quantitatively and qualitatively into the two daughter cells, which are divided from the parent cell. Okay, so it was first observed by Strasburger in plant cells in 1870. Then in 1879 in animal cells by Bovary and Fleming. Okay, so mitosis occurs in the formation of somatic body cells. Okay, that is non-reproductive cells. That is called somatic cell division. Whereas in the reproductive cells or germ cells, meiosis is observed. Okay. So in here in the mitosis, we have early prophase, then late prophase, then metaphase, anaphase, telophase. After telophase, the cell division happens and again it enters into the interphase. Okay. Okay, I will stop here. So I will continue tomorrow. So are there any doubts?
No, ma'am. From tomorrow, we are starting from mitosis. Yes?
Yeah. So cell division, that is mitosis. So all the three phases I have explained. No? So because if I start now, like the time won't permit. So again, I have to stop in the middle. Okay, so freshly we will start from tomorrow, from mitosis. Okay?
Okay.
So, okay. If there are no doubts, let's call it a day. Okay? Thank you.
Thank you, ma'am.